drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Can you start off by just telling everyone a little bit about who you are and what your lab studies?
Sure. So I'm a professor at the University of Toronto, and I'm in the Department of Nutritional Sciences, which I'm sure we'll get to the significance of that a little later on. And I'm a neuroscientist, maybe an old breed of neuroscientist, which you might call a neurochemist. And we're really interested in brain neurochemistry with a focus on the lipids. You might know this, but if you exclude water, your brain's pretty much half fat, and that's almost as fat as your body fat. So we're interested in, how does the brain get to be like that? That comes back to part to nutrition. And then the ultimate question is, why is it like that? And that's, a broad area. So we use a variety of model systems, including human clinical studies, to try and get at the answers to some of the questions we have.
Mm-hmm. And just to give us a bird's eye view, like what components of neurons and the brain generally is fat used to build, and how does that compare to the rest of the cells in our body? Do neurons have more fat than other cells or less fat? And is it just the cell membrane, or are there other components there that are made out of fat?
Yeah, so there's a few things there. From a lipid chemistry perspective, if you stand out far enough and you look at a cell, they're quite similar in that the phospholipid membrane is phospholipids, which is a lipid, and then there are fatty acids in there. They vary from tissue to tissue. The brain's a little unique, and we can get into that. But the brain's also a little unique in that lots of our tissues would also have a fair amount of triacylglyceride or triglycerides, kind of a storage fat that people would think of either that they're using it for energy when they're exercising, or it's the thing that's, putting on a few too many pounds. Our brain doesn't have that. The other thing with fats, though, is they're in this membrane, but they also can come out of the membrane. And in that respect, they're involved in a lot of signal transduction. So we study them at kind of both of those levels because there actually are some big questions on how the composition of the membrane affects the amount of fat that's released for signaling
Hmm.
afterwards.
Hmm. Okay, so they're not just structural things.
No, no. If you picked up a textbook in biochemistry from about 1975, the year I was born, Leninger would say they keep you warm in the winter. They have structure. They're used for energy. Something I'm probably forgetting because I'm trying to count to four on the spot right now. But they have no, unlike nucleic acids and proteins at the time, they have no ability to share information, and that's not true, right? In 1982, the Nobel Prize was given out for the discovery of a molecule called prostaglandin E2.
Hmm.
Back step a little bit. Prostaglandin E2 is made from cyclooxygenase. That's the enzyme that aspirin inhibits.
Mm.
And if you step back a step further, that comes from arachidonic acid, which is a fatty acid derived from essential fatty acids. And that, classically was thought to relay information about inflammation. Arachidonic acid is one of the fatty acids that's, in the neuronal membranes and the microglial membranes and a lot of the different type of membranes. And it, has a fluidity and a structural function, but it also gets released in response to signals and relays further signals. So
Hmm.
they have a very important role structurally that's been, probably the focus of 40, 50 years of research. But more recently, we're getting into these nuances of their derivatives, sometimes called bioactive lipids, and their role in signal transduction.
Hmm. Yeah, I definitely want to get into a bunch of that stuff at some point. Stepping back a little bit, so when we think about fatty acids generally, people maybe are most familiar with seeing some of these things on nutrition labels and things like that. There's different types based on their chemical structure. You've got saturated, monounsaturated, and polyunsaturated fatty acids. Can you give people just a general sense for what the major differences between those types of fatty acids are?
Yeah, so you nailed it there. We've got saturated fatty acids, which the if you can picture a structure in your head right now, you've got a series of carbons connected to each other and a carboxylic acid at the end. When they're in a phospholipid, they're no longer an acid because there's an ester linkage there. And saturated, at least when I visualize a fatty acid, I don't do this innately. I just see the carbons, but there's hydrogens all there, right? And so saturated will often be referred to as there's no double bonds. True, but not quite accurate. What it means is saturated with hydrogen, so therefore there are no double bonds. And if you had a double bond in, you've removed hydrogen, so it's now unsaturated or monounsaturated. And then if you've removed multiple, you get these polyunsaturated ones. And the brain's a little unique in this respect, in that there's one of these polyunsaturated fatty acids called docosahexaenoic acid. Doco means 22, hexa means six. And so it's 22 carbons long and it's got six double bonds. I'll call
Mm.
it DHA. And that one's, really unique in the sense that it's highly abundant in neuronal membranes. And we find it at the level of the synapse, quite enriched. In some specific phospholipids, it can reach 40 to 50% of the composition of that phospholipid. So there's spots in the brain where this molecule just lives, so to speak. And that's unique composition. So we don't see that in the liver and the muscle. There's DHA in those tissues, but not quite at that level. And it begs the question, why is that? How did
Mhm.
it get there and why is that?
Mhm. And so, in terms of the saturation, we're talking about how many double bonds exist between the carbons in sort of geometric terms, what does the level of saturation mean in terms of the overall shape of the fatty acid, whether it's sort of straight or bent or highly curved, and what significance does that have?
So two things. So in a simple model system, it's easier to pack saturated fatty acids on top of each other. You can make them more tight. So we talk about the membrane being more rigid or less fluid. And the implication of that is that proteins or receptors or anything in the cell will have different patterns of movement, maybe slower patterns of movement in a more rigid membrane. But there's also something, and then you increase the unsaturation, the membrane becomes more fluid and things can move around a little easier, so to speak, in a very simple model system. But it's also very important for signaling because a lot of these molecules like prostaglandin E2 essentially require oxygenation or hydroxyl groups to be added to them. And those are typically added at the sites of unsaturation. So you can't make a prostaglandin E2 from a saturated fat like palmitate. Palmitate 16 carbons, but even if it was 22 carbons, we call it, or 20 carbons, we call it a rachitic acid. Similar to arachidonic. You probably heard that even with my little French Canadian accent. Hopefully you got the difference. You can't add hydroxyl groups to a rachitic acid, but you can add them to arachidonic acid because it's got the double bonds there, and that becomes very important in the signal transduction afterwards. So membrane fluidity and signal transduction are reflected in this saturation or unsaturation and the various indices around those.
I see. So the amount. So if we imagine a cell, we have the cell membrane. There are proteins in the cell membrane, like receptors and different things, and they don't stay in one spot. They're kind of moving around in this membrane. And the membrane, I don't know if we think of an analogy or something. I could imagine wading through a pool of water, maybe, or a pool of molasses, and that would maybe, if the molasses is going to be harder to wade through, I'm going to move around a little bit more slowly. That would be akin to saying the membrane has, a higher or lower saturated fatty acid content, and it's more or less fluid.
Yeah, so I like that, and I'm going to steal that from you because it's better than how I teach it. It's perfect. The one thing I'll say though is that's, it's a little more complicated than that because if you, it depends on the fatty acid that's beside it a little bit. And then things can get really complicated because the membrane's made up of phospholipids, and there are specific types of these phospholipids. Predominantly phosphatidylcholine. There's another one, ethanolamide, and there's subtypes of those. And then there's inositol and serine. And for whatever reason, they have their own biases or affinities. So you kind of can't put two palmitates in a phosphatidylserine in reality, right? It's usually got one of these DHAs in one position and something else in another position. So I can't really explain all the biophysics of this, but sometimes you'd say, if I put a palmitate, I'm trying to do this in my head with something with one double bond might be much more rigid than something with two double, it doesn't add up 1 to 1 to 1 for every. You can't count the number of double bonds and say this is the fluidity of the membrane because it depends on some interactions, I guess is what I'm trying to say.
I see, but the sort of profile of fatty acids in the phospholipids of the membrane is going to determine how fluid that membrane is, how easily proteins and stuff can move through it, and presumably the fatty acid composition of membranes in the brain and elsewhere in the body, presumably that changes depending on the fatty acid profile of our diet, or is that not true?
Yeah, and that's why I'm a nutritional neuroscientist, right? And so we should take a step back and go, there's another way to classify these fatty acids. We've called them saturated, monounsaturated, or polyunsaturated. We can also call them essential or non-essential from a biological function. Okay, we'll separate. I think they're all essential biologically, but dietarily essential. And that brings us into the field of nutrition. And so these saturated fats and these monounsaturated fats, we can make, okay? We can eat them in our diet and absorb them directly. Palm from almost any sort of fat is going to have palmitate in it. oleic acid, one of the monounsaturated. Sounds like olive oil. It's famous for olive oil, but it's in all kinds of things. You can eat them, or you can use glucose and make them through lipogenesis. And we have some enzymes that can make oleic acid because that double bonds in a specific position, and we have an enzyme to do it. Then you get into the polyunsaturates, and there's a couple minor exceptions to this that I don't think we need to get into, but there's roughly two types. There's the omega 6s, and then there's the omega 3s. And we eat in our diet essential precursors. So there's two of them, and the naming's awful here. You got to pay attention. There's linoleic and linolenic. It's sometimes I trip it up. Like so,
Yeah.
linoleic comes from the omega 6 side, and linolenic, sometimes called alpha linolenic, comes from the omega 3 side. And they have a double bond in a specific spot, one in what's called the omega 6 position, one in the omega 3 position. And if you're in organic chemistry right now, you're going to be confused because you start naming the fatty acids from the carboxylic acid end of the molecule. In nutrition, we start on the other end, the methyl end. So if you say the bonds in the 3 position, you got to tell people if you're a nutritionist or a chemist, right?
I see. So if someone's not a chemist or nutritional chemist, the names, you've sort of said this in multiple ways already. The names, these long, complicated names, and the numbers like omega 6 versus 3, that's just referring to like how many carbons or the position of the bonds. It's referring to something about the chemical structure.
Yeah. So typically, there's the name kind of usually referring to where it was discovered. Or we get into Greek to make it a little more simple kind of thing. But even some of the Greek some of the names like Dacosa or Penta something, that we have a common name for them, usually based on where they're discovered. But these positions are telling us, Dacosa hexanoic acid, there's 6 double bonds. There's nothing in that name that tells us where the double bonds are, and you'd have to know that the first one's an omega 3 because that's an omega 3 fat. But anyways, going back, we've got these 18 carbon precursors that we eat in our food, right? In a variety of food sources. And then we can take them in our diet, bring them to our liver. We can make them a bit longer, and we can make, in one case, that arachidonic acid from the omega 6s and DHA and some other steps in here. And we can eventually get them into the brain and into the membranes. The other thing to make it just a little more complicated is you can either make DHA or you can eat it directly, and it's famous for being in fish. So you have a choice. You can either eat it from fish or you can get the precursors and get it, but you have to eat it, okay? There's no glucose coming in here. There's no amino acids. There's no lipogenesis. You have to get the 18 carbon precursors started, and that's why we call them nutritionally essential, which, doesn't necessarily speak to how biologically essential they are.
Yeah. So all these things are essential biologically. Our body needs all of them, saturated, monounsaturated, and the different polyunsaturated fatty acids. But as you said, the saturated and the monounsaturated fatty acids are not nutritionally essential. So our body can make them, but these omega 6 and 3 fatty acids, the polyunsaturated ones, are nutritionally essential. We have to get them from the diet. Does that tell us anything about their significance or how they're used? Why is it that some of them our body can make and some of them we can't?
Yeah, so it's a good question. And, that reminds me of my PhD exam. And I was very interested in DHA in my PhD exam. And so one of the neat things about probably in humans to some extent, but definitely in animal models, if you remove it from the diet, you can lower the levels in the brain. And then we can use that to start to study its biology and its physiology, right? And so that's really exciting because there's wiggle room with this fatty acid. You can have higher levels or lower levels. And does that matter? Okay, so we're really excited about that. But the question came to me as a young PhD student from the examiner: palmitate, it's almost impossible to change in the brain. Okay, and we've done some work on this. We've published some of this recently. It's so rigid. You can remove it from the diet, brain levels stay the same. We make diets that aren't even, don't even model what humans would eat. Levels basically don't change in the brain. And so the question the examiner said to me, well, doesn't that mean it's more important? because there's tight biological regulation on that. So it's a good question. At the time I did my PhD a long time ago, we didn't quite have the tools to answer that. I still think we don't have the answer, but now we've got biological tools with knockouts, and they tried to knock out the enzyme fatty acid synthase a long time ago when I was a grad student, and it was lethal. But now with tissue specific knockouts, I think we're going to get a better idea, and the lipid nerds can sit down and say this molecule is actually more important for that than that one. But the lipids have been really tough to study, largely for this reason. You can't knock out a lipid. There's no gene
Hmm.
encoding for palmitate. There's genes encoding for its synthesis, but there's a variety of them, and there's no gene encoding for DHA, right? So you can't knock it out. So we've struggled a bit compared to some of the other fields not having those tools available to us.
Hmm. Interesting. We're going to spend most of our time, I think, talking about brain lipids and stuff to do with brain health and disease. Before we get there, I want to talk about just some basic fatty acid metabolism and probably just talk about what the liver is doing here. So, I think you mentioned briefly before that the liver it can metabolize fatty acids. It can make them longer. It can change them in different ways. I believe it can sort of make them from scratch or process and change some of the ones that we can't make from scratch. But can you give us just sort of a basic crash course in fatty acid metabolism? What are these terms: desaturation, elongation, and oxidation, and what do those things mean?
Yeah, maybe we'll start from the mouth, right?
Okay.
Most of the fats we eat are in the form of triglyceride. I say triglyceride, a chemist would say triacylglyceride. Sometimes we call them tags. That's usually three fatty acids bound to a glycerol molecule. You eat others. You eat some phospholipids, you eat some other things, but triglycerides 98, 99% of your diet. There's some enzymes called lipases that start actually at the lingual lipase and kind of make your way down, and they essentially break the fatty acids off of usually there's three positions. They break it off in the 1 and 3 position, typically leaving the 2 position intact, which might be important for some things in development. There's that one molecule DHA in human breast milk is in the 2 positions, suggesting it's protected, right? And it's important for neurodevelopment. It's just kind of a neat thing. We might go there. And then you take these fatty acids and you bring them into the intestine, um, and you basically repackage them all. So you've broken them all down on one side, and you bring them all back in and you repackage them all into a chylomicron. You send them out. They drop into the lymphatics, and they go to a variety of places, including the liver. A notable exception, and you have to put them in these chylomicrons because they're not soluble in your blood. And one of the things these chylomicrons, which are essentially lipoproteins, do is they help them solubilize them in the blood so we can transport them around, so not transplant them. But there are exceptions. My bias is I start thinking of fatty acids at about 14 carbons and longer. It's totally not true. There's shorter ones, and there's a lot shorter ones. And as they get shorter, they become more water soluble,
Mm.
even though we'll call them a fatty acid or a lipid sometimes. And those ones have different absorption mechanisms instead of through the lymphatics, through the portal, and they don't require chylomicrons. So keep that in mind. And then a fatty acid kind of comes into a cell, almost any cell with some differences between tissues, and the cell can say, hey, I'm going to take you and store you into triglyceride, something we don't really do in the brain, but you do in the liver, you do it in your muscle and others. Or I'm going to throw you into the membrane, or I'm hungry, I need some energy, and I'm going to oxidize you, beta oxidize you. And I'm going to take you through, a series of steps, break you down to acetate, and we'll put you through the TCA cycle, and we're going to eventually make ATP out of this, and it's quite a rich process. If you've got double bonds in you, I can still do that, and there's some minor exceptions to the pathways. But just one thing for your listeners to be aware of, we can also auto-oxidize you. And
Mm.
that's when oxygen reacts and makes oxidative products, which sometimes are considered bad things, but they might also be signals for other cells that something's going on here. So don't just like to call them bad. And so they get into the liver, and then eventually they get into the blood and to the adipose tissue in a variety of ways. And then they, circle past the brain. And I think in the human, the blood-brain flow is about 2 seconds, something like that. It's just under a second in a rodent, and they have to cross the blood-brain barrier and eventually get into the brain, is assuming that's where they're going. And there's a lot of debate in that, how that works in our field. Okay, I'm part of the debate, but I'll try and give you two sides of it. The fatty acids in the blood can be in lipoproteins. There's a variety of them. LDL being a famous one people might know about, but there's others. It could be as a free fatty acid or as what's called a lysophospholipid, which is a phospholipid with a fatty acid missing on it. And so my colleagues a long time ago could make artificial membranes with no proteins in it, and they could see free fatty acids cross those artificial membranes. So we realized that fatty acids might not need a transporter to cross, okay? We came into the field and we started knocking out the candidate lipoprotein receptors. Didn't seem to do much, so they didn't seem to be quantitatively a major role. These lysophospholipids can also cross. And so now we've been debating, well, are they crossing just like free or are things helping them? And this has been a massive area of confusion for our field because we named a whole bunch of proteins fatty acid transport proteins. Pretty clear by the name what they do. You have a membrane, a fatty acid on one side, you increase the amount of fatty acid transport proteins, FATPs. Fatty acids go through it more quickly. Looks like a transporter, smells like a transporter. Is it a transporter? No. They're acyl-CoA synthetases, which are molecules that take a fatty acid when it's in the membrane or other places, and you put a big coenzyme A on it. And coenzyme A is massive and it's water soluble. And that does two things. It sucks it out of the membrane, doesn't let it flop back across, and makes it kind of water soluble. But the problem is, if you look at this, because it's facilitating the uptake, it looks like a transporter. I like to compare it to glucose and hexokinase or glucokinase. Glucose goes through a GLUT4 transporter, gets 6-phosphorylated, and essentially disappears so more can come back in. But if you looked at hexokinase activity or glucokinase activity, you'd say, oh, that's facilitating the uptake. It's a transporter. Until you get really close and you realize it's metabolizing the glucose, allowing more to come in. And a lot of our proteins that are called fatty acid transport proteins are really involved in the metabolism of fatty acids. And papers, and you could Google this right now and you'd say you're wrong 500 times. Everybody's calling them transporters, but this is an area of debate. And there are some others. There's one called CD36, and there's one called the MFSD2A that probably works on the lysophospholipids to help bring them into the brain. And eventually there's a series of enzymes that help them kind of get where they're going. And I almost can't remember what the question was now.
Well, it sounds to me like basically one important part of what you just said, if I'm hearing you correctly, is that the type of fatty acid we're talking about in terms of its geometry, the number of double bonds, whether it's saturated, unsaturated, as well as its overall length, if it's short, medium, or long, those things are going to dictate how it gets absorbed and transported throughout the body.
Yes.
And so one of the key steps in fatty acid metabolism, and one that certain corners of the internet and the world talk about a lot, is oxidation, as you mentioned. So fatty acids can become oxidized. How many double bonds they have is going to tell us how many places can become oxidized. And you said something that was kind of interesting. You said oxidation can have a negative effect. It can produce, and my understanding is they can produce things that are toxic, oxidative byproducts, but you said they could also produce things that carry information or act as signaling molecules. Can you say a little bit more about that and the role of oxidation?
Yeah, so two things I'll say about oxidation. One is that a little bit a better model in the periphery is that we think some cells can recognize immune cells, these oxidative products, and then I don't want to overstate it, but like chemotraxis, and they go there and then they do things. So they give off a signal that's not always bad. Clearly, they can destroy membranes and not help. One of the mysteries that I'm going to throw out right now is if I were designing a brain or a body, I would keep these polyunsaturated fatty acids away from the sites with the most oxygen uptake, right? Oxygen's what's going to oxidize them. I'd put them in the opposite places. What do we see? The exact opposite, right? There's a positive correlation between oxygen consumption of a tissue or brain region and the polyunsaturated fatty acids, this DHA with six double bonds, that would be most susceptible to autooxidation. They're in the same place. You can ask me why. I'm going to tell you I don't know,
Hmm.
right? Nobody knows this. And I'll bring it back a little further. It's really wild because it jumps across animal kingdoms in that two of the richest non-mammalian sources of DHA are the hummingbird breast muscle, which has massive oxygen consumption,
Hmm.
and the little muscle that I can't name on the tip of the rattlesnake tail that beats really quickly, that apparently also has a lot of oxygen consumption. They're rich sources of polyunsaturated fatty acids that would be highly susceptible to oxidation. So PhD thesis waiting to happen. I don't know why that is. I've had conversations with people. We have hypotheses, but nothing I'm compelled to tell you right now. Like this is the
Hmm.
one
Hmm.
that we're betting on, kind of thing.
Um, another way that oxidation gets discussed is so these things can become oxidized in the body as part of the chain of metabolic events that's happening. They can also become oxidized outside of the body before we consume them. So
Yeah, yeah,
one
yeah,
place people talk about these are like
yeah.
French fries or something at fast food. It's basically you're boiling these long chain fatty acids. They become oxidized before you even put into your body. Does that have an impact in terms of how they're used by the body or their toxicity or anything like that?
Yeah, so I'll, if you Google this in the internet, you'll get the answer, and this is the devil's oil, and this is the root of all evil while everybody's dying. I don't think that's the case. But we're starting to do research now, and there's somebody named Amir Taha at the University of California, Davis, and others are really interested in this question because they're starting to, the first question, these oxidized lipids, for a variety of reasons, would have a hard time getting absorbed, okay?
Hmm.
Turns out they're absorbed, though, just in small quantities. We don't know the details of how they're absorbed. Like I can't draw you that. You can't open a physiology textbook and find that. You really can't even find a paper and find that. We assume it's similar, but we might not quite be right. But they're absorbed. They seem to be absorbed in relatively small quantities. So the body clearly has some mechanisms to try and keep them out or get rid of them, which might relate to their toxicological properties. But yeah, you're absolutely right. They can be found in foods,
Hmm.
especially foods that have been exposed to oxygen or
Hmm.
left out. Their levels accumulate. Um, Sometimes fats, and there's some little details here. I don't want to put my neck out too far, but smell bad, and it's not exactly clear to me at the most minute level if it's just oxidized fats or if we need amines in that mixture to
Hmm.
make things smell bad, fish being a great example. And that these are also cues for us not to eat these foods,
Hmm.
that something's wrong with them. And then the debate we have there in the field, are they the toxin or are they the canary in the coal mine? For something else, or are they both, right? And so we're working on that. A lot of people are working on that. But there's clearly, evolutionary program signals where we tend not to like oxidized lipids from a nutritional
Hmm.
perspective,
Hmm.
or
So
foods with oxidized lipids in them.
So based on how we prepare foods, with a lot of the vegetable oils and stuff that get heated, there's a lot of oxidized lipids out there that we put into our bodies. It sounds like you're saying most of those oxidized fatty acids don't get absorbed, and that it's probably a good thing that we only absorb a tiny little bit. One place my mind goes there is I assume that's under sort of baseline conditions. A healthy gut doesn't absorb a lot of those. What if there is some kind of a gut dysbiosis and there's like a leaky gut? Does that mean that some of those things can get in more than you'd want them to get in?
Yeah, so great question. We don't know yet. Nobody's done that yet.
Yep, yep.
At least to the best of my knowledge, I know a couple people working on this. And when you start these studies, you start simple, right? You don't go with the more complex pathophysiology. Very reasonable hypothesis. My guess is probably, but I don't know that. Interestingly, the infant, which would be another cause for concern with, if I can use your term, leaky gut, usually is drinking human milk, and that would not be a major problem in human milk, right?
Mm-hmm.
Under normal circumstances. So Um, Great question. I don't know the answer to that one. I
Got
think
it.
so, but I'm not sure.
And before we dive into the brain more, one more question about the liver I want to explore is, does the fatty acid composition of our diet have an influence on things like fatty liver disease and how much fat actually accumulates in the liver based on, based on which fatty acids we're actually giving to the liver?
Yeah, yeah. So it does. And it's not always clear if the dietary fat is regulating that. So I think excess calories in general can contribute to a fatty liver, and then disease states as well. But when we look at fatty livers, they tend to be more saturated and maybe monounsaturated. So if you took a fatty liver sample and you ran it on an instrument, maybe I'll just we call it whatever, a mass spectrometer, or what we call the GC, you'd see that it's biased, and it's palmitate and it's oleate. But it's not 100% clear to me if those were the dietary fats in the liver that accumulated there, because those are the two you can also make from lipogenesis.
Hmm.
And maybe somebody knows that answer, and I'm just unaware of those studies. But it seems to me it could be either of those two or a combination of those two. Nobody's reporting fatty livers with omega-3 fats, right? Like we don't
Mhm.
we don't see that in the literature. If anything, there's a small literature that some of the dietary omega-3 fats could maybe mitigate or attenuate the effects of the fatty liver, or shrink it just a little bit. Not a sledgehammer, but a little effect there.
Mhm. And then, when we start to think about the brain and we think about the fatty acids in the brain, how are they getting there? Are some of them made inside of the brain? Are they primarily made elsewhere? Then they go through the bloodstream, across the blood-brain barrier into the brain. How do the fatty acids in the brain get there?
Yeah, so this is, I'll start off simple, and then I'm going to confuse everybody, because I'm a bit confused, and we just published a study on this, and the results were wild. So these omega-3s like DHA enter the brain through one of those kind of mechanisms we spoke about a little bit. Then it's really wild, because there's another fish oil, if I can call it, omega-3, called EPA, eicosapentaenoic acid. We haven't talked about it. It's barely detectable in the brain. So if I showed you like a readout of one of these chromatographs, there'd be a little something that you might call noise and I might call a peak, and we probably agree to disagree on. So it's so low in the brain you can barely detect it compared to the other things that are really clear.
Hmm.
What we found, though, that was wild, is it gets into the brain at about the same rate as that one DHA, roughly the same, but you almost can't find it in the brain. So it's metabolized, maybe I should use the word catabolized, very rapidly. How fast, I'm not exactly sure, but probably within a minute, most of it is either destroyed or turned into something else. So they get into the brain, but my point here is that the brain itself can help regulate its own composition.
I
Okay,
see.
it's things are getting in there, and you'd predict to see them in there, but you don't find them in there, so the brain's doing something with them. Okay, and then we've got this molecule. Let me back up a step. So if you look at the brain, uh, usually the most abundant peak, and I use the word peak interchangeably, unfortunately, with amount, is oleate. And then you've got one called oleate, because this 18 carbon with one double bond. Then you've got one called stearic, found in steers. It's 18 carbons, no double bonds. Palmitates right in the running there, sometimes second, sometimes third. And then you've got DHA probably about fourth, and this one arachidonic fifth. And then things, with the odd exception, really drop off a lot, right? So they're there, but they're an order of magnitude lower. What we've just discussed probably covers more than 80% of what's in the brain, just those few. Um, and so some of them get into the brain, some and get, we see them there. Some of them get into the brain and we can't find them there. And then there's this molecule called palmitate that I told you is very hard to change in the brain. Okay. And it turns out that the brain has these enzymes that can do lipogenesis.
Mhm.
So the brain can make palmitate. Okay. Um, there's a lot of palmitate in the blood. And it turns out that it also comes in from the blood and enters the brain. So the brain doesn't say, I'm making you stop coming in. It lets you come in as well. What we did that was a little wild and a little surprising is we gave rodents diets essentially palmitate free, not quite, or very high palmitate and in the middle. And what we found was that even if we kept these animals on these diets for a long period of time, we couldn't change brain palmitate levels. I hinted that it never changes, right? So it's quite stable. And what we found is that the using isotopes, if I can call it that there was more when we removed palmitate from the diet, the palmitate in the brain was more synthesized, right? From the carbohydrates, the glucose. Makes sense. What doesn't make sense is we did a lot of work on this and we found that it was actually the liver that was synthesizing it and sending it to the brain. And so the brain has the machinery to make it, okay? It can take it up from the blood, but when there's none in the diet, the liver upregulates its ability to synthesize it, not the brain. The brain's got I don't want to overstate it, but it really looks like a static level of palmitate synthesis or lipogenesis going on. And if you give it more, it doesn't downregulate. If you give it less, it doesn't upregulate. It relies on the liver to do that. And I'm not sure why that is, but cancer cells do something very similar. They have a very critical level of lipogenesis. And if you block that with a drug that inhibits it, you kill them. And then the textbook experiment is, well, I'm going to stop it from making palmitate with this drug. So if I add palmitate back, I'll rescue it, right? You can't rescue it. Cell still dies. And then the wild thing is there's tons of, I don't know why it's making the palmitate because there's tons in the blood and the vasculature it could just take. And what it makes, it secretes back out into the blood. So it's kind of useless. Like we're missing something, right?
Yeah.
And the brain looks a little bit like breast cancer, and the studies were MCF7 cells, which are breast cancer cells that I'm thinking of. But the brain looks a little similar to that. Like it has to make palmitate for some reason, and it doesn't change that, right? So we're missing a little something going on there, and it doesn't seem to change its regulation.
Interesting. So the brain has this sort of constant level of palmitate, a saturated fatty acid synthesis. It never really changes. But the amount of palmitate made in the liver will change based on your diet, and that's going to influence how much gets into the brain from the outside. But somehow through all of those changes, the brain is sort of like holding the palmitate levels constant.
Exactly. And so I don't mind hypothesizing too much, but I think sometimes we can measure palmitate and that becomes that's the point of lipogenesis. That's why we call it lipogenesis, lipidgenesis. But there's a lot of little reactions in there that balance things like NAD pH and NAD ratios. And I'm without data speculating that might be also very important. And maybe we should have called it, NAD regulation and genesis.
So maybe the palmitate synthesis is sort of a byproduct of something else that's going on. Yeah.
It looks like a good hypothesis in the cancer cell that secretes it back out. In the brain, I have to add on another layer to say, and the brain's evolved a mechanism to store it in the membranes, right? Why waste it? We'll put it in the membrane. I got to add that little part of it.
Yeah. And so I would imagine that in the brain, from a structural perspective, obviously you've got your phospholipid bilayer of the neurons themselves is going to be made, and other cells is going to be made out of fatty acids. Presumably it's also a component of the myelin. And I would guess that maybe there are some differences there in terms of which fats get used for cell membrane versus myelin.
Yeah, a big difference actually, and great for bringing this up. The myelin is really enriched in palmitate and another one called oleate. So it's relative to the, if I can say the rest of the brain, it's quite rigid, or it would not be a very fluid membrane. Very biased in that respect.
Because it's got mostly saturated, monounsaturated, and very little polyunsaturated.
Yeah. Not none, but very little. So roughly speaking, if you look across the whole brain, a molecule like DHA would be 10 to 15%. It would drop down to about 1% in the myelin. So when people say the brain composition of this is this, it does vary a lot by cell type and architecture of the cell, with the palmitate and the oleate jumping up even higher in uh,
Hmm.
the myelination.
Yeah, and as you mentioned before, these fatty acids, they're not just used for structural reasons. They're not just building physical structures. They're not just used as an energy substrate to used to make ATP for the cell. They can actually serve as signaling molecules themselves. And I'm wondering if you could start to tell us a little bit about that. So in the brain, how is some of the signaling happening? How is it affecting synaptic transmission? What are some of the major things we know about how some of these fatty acids are actually used for informational purposes?
So two things, and one I think you've covered nicely earlier is by changing the membrane fluidity, they change, how the receptors, whether their spatial orientation or their movement, so they can have those effects. But the other thing we've known for some time now, and I'll use dopamine as an example. Dopamine is G protein coupled, the D2 receptors G protein coupled to an enzyme called phospholipase A2. Okay. Uh, Phospholipase, it's like phospholipid lipase, like the digestion lipase we were talking about earlier. A2 is when you look at a phospholipid, there's two positions. There's the one position, the two position, and then the three position kind of doesn't count because that would be a choline or an ethanolamide. But in the one and two position, there'd be a fatty acid. And usually, or at least a high bias, the two position is a polyunsaturated fatty acid like this DHA or that other one, arachidonic acid. And so dopamine binds to the D2 receptor. You've had a lot of neuroscientists on here. Everybody's interested in what happens after that. One of the things that happens after that is phospholipase A2 gets activated and it releases the arachidonic acid, typically, depending on a few little things. And that arachidonic acid comes out. Some of it gets converted to what we call oxylipins now, which would include this prostaglandin E2, but this is a field that's blown up and that there are many, many of these molecules. And then it does something. And in some cases, it might be, it's relaying the signals of dopamine. But we haven't worked that out systematically yet. So, the work that was done on olfaction, how they systematically work that out. We need to do that with these molecules because there are hundreds of them and they're being released and they're doing something. And I'm not going to do a very good job right today in telling you what they do, right? I'm going to write that as a grant. But one of the wild things is that arachidonic acid that comes out, about 97% of it just goes right back into the membrane. We call that the Lanz cycle, named after Bill Lanz, and we don't know why that is either. So you take a whole bunch out, you lose about 3%, and you bring 97% right back into the membrane. And we do that with other fatty acids as well.
Mm-hmm. What about things like, so I know that certain fatty acids that are ultimately of dietary origin, they connect to endocannabinoid production. So can you just remind listeners what are endocannabinoids and what's the connection here between dietary fatty acids and endocannabinoids and what they're doing to regulate synaptic function?
Yeah, and we'll go back maybe a little bit to some of these oxylipins. So we've got fatty acids and then we've got fatty acid derivatives, and we use these terms like bioactive fatty acids, and there's a lot of different types, some we haven't talked about. Ooh, I don't think I've ever had to define endocannabinoids on the spot, but they're essentially, people found the CB receptors, the cannabinoid receptors, CB1, CB2, which THC binds to, and then it was obvious, hey, they're not made for marijuana. There's something endogenous in there, and the first big hit was a molecule called anandamide. And anandamide is actually derived from arachidonic acid. So it's an ethanolamide attached to arachidonic acid, and that's anandamide, and it binds to the CB receptors. Huge discovery, really elegant stuff. And that really helped us understand its role in appetite regulation and a lot of the things that people would be familiar with. Then it turns around, the story gets a little more complicated because arachidonic acid is an endocannabinoid because it binds to CB receptors. It's to a chemist, it's an ethanolamide. It's just what you would call it, okay? And then we find that I think all of these fatty acids, or every one we've looked at, can make these ethanolamide derivatives. So if you're a
Hmm.
chemist, you're like, I got this, no problem. Okay, they all make them. There's these enzymes. There's a series of enzymes that do this, and you make them. Where the biologist maybe gets a little annoyed with this is they're not all endocannabinoids. Endo meaning endogenous cannabinoid, in that they don't have affinity for the CB receptors. Maybe they bind to PPAR, maybe they bind to something else. Some of them we don't even know yet what they do. But they're involved in a lot of the synaptic transmission as well. They're produced locally on demand, especially in neurons. And they really, seem to be very important for synaptic plasticity and models of memory and learning. And interestingly, some of them you can change a little bit with your diet.
Hmm.
And one thing I think we have to be careful with nutrition is if you look at something and it's one's higher than the other, I don't know if the one's higher or the one's lower. Okay. It's just always a problem we have in nutrition. If I say this one's higher, your listeners should say, well, how does he know the one's not lower? Fair point. Okay. We just use the language to get us by. So, but we can change the levels of some of these with diet. One of them is derived from DHA, and we would call it DHEA, which is terrible if you study hormones, because it's
Yeah.
not that one, but it's
Yep.
ethanolamide, not the hormone, and that is really important for synaptic plasticity. And at least in animal models, we can change this level and change those functions. So there's a whole cascade of those molecules. My lab doesn't discover molecules, but the only one we've ever discovered is one of these longer chains. So DHA can even get bigger. From DHA, it's called THEA. It's got a really long name. And we found that during ischemia, that one gets released. And if you add it onto neurons, it can affect synaptic plasticity as well.
Okay, so some of these dietary fatty acids, like in particular linoleic acid, omega-6, polyunsaturated fatty acid, you get things like that in the diet. We eat it. Those things can be turned into arachidonic acid or any number of other fatty acid derivatives, and some of those act directly as endocannabinoids. So some of those fatty acids are literally behaving almost like neurotransmitters.
Yes, I want to make an important distinction here, okay? We've got a cell membrane in the brain. It's about 10,000 nanomoles of a molecule like arachidonic acid per gram of tissue. Okay. I'm using 10,000 to keep the math easier. When that phospholipase releases it into the free form, it's about 1 nanomole per gram. Okay. So there's a 10,000 fold drop, roughly speaking, there. And then when we get into these other bioactive mediators, including prostaglandin E2 or anandamide, which is coming through a slightly different pathway, we drop at least another 10-fold from the one, if not 100-fold. Okay, so why am I telling you this? Well, we've got a bit of a mystery in that we think nutritionally speaking, and we have evidence, we can regulate the levels of those mediators. Okay. So if I give you a diet very, very low in omega 6s, we will lower some of those molecules in your brain.
Mm-hmm.
That makes no sense to me because we still have 9,990 nanomoles in the membrane. And I've 10,000, I've taken away 10, I still got a ton left. And yet the brain's saying, oh, something's wrong, I can't make these. Okay. And that's a mystery to me why that is. It's like saying, you have $1 billion in your bank account. I take away, 10 million from you, and you're like, I can't buy a coffee at Starbucks anymore. Like it doesn't make sense, right? So we're missing something, and this is a bit of a mystery. Why are there so many of these in the membrane? Why can we change the membrane? But the changes in the membrane are not going from 10,000 to 10. They're going from 10,000 to, 9,990. And yet we're still changing the levels of these mediators.
Hmm.
And we can show there are consequences to that.
Hmm. Um, so when we think about dietary fat intake and the effects on the brain, we've already started to put some pieces together. A lot of these lipids act as signaling molecules. They're made from our dietary fatty acids. So now we can get thinking about like how our diet can influence things like endocannabinoid levels or levels of other fatty acids that are, behaving almost like neurotransmitters in the brain. So if we think about the typical American diet is going to be getting way high levels of omega 6s, which are going to give you, arachidonic acid and the endocannabinoids. And, if you're getting sufficient omega 3s, maybe you are, but a lot of people might even be deficient in omega 3s. Can you start to talk about what the typical American diet means in terms of, effects
Yeah.
on the brain via endocannabinoids and some of these other things?
So two things we're going to do here. I'm going to just give you some numbers so you can see the perspective. And the first theme is going to be slowly and buffered. Okay. So I'm going to use DHA as an example. I could use arachidonic acid as an example too. But DHA, somewhere around the third trimester, it starts to accumulate in the infant brain. Um, you know, Postnatally, it accumulates a little bit. People are going to disagree with me on this, but somewhere around 2 years of age, it plateaus. Okay. What level does it plateau as? And I'm going to use slight rounding just to keep the math a little more simple. Okay. I'm going to say it plateaus at 5 grams. So you in your brain, you have 5 grams of DHA. I think you probably have about 3.9, but we're going to say 5 just to keep the math easier. So you've got that in your brain, and everybody's like, oh, 5 grams, that's a fair amount. Yes, it is. Then what we've been able to do using PET imaging techniques, so with these carbon 11s, is we've been able to image how much DHA is going into your brain, okay, every day. And the answer is about 5 milligrams of DHA. And so I picked those two numbers so people can kind of do the napkin calculation at home. And it roughly means the DHA that's entering your brain is going to last for about 1,000 days, about a half-life of about 500 days. Okay. So that's one of my first themes, slow. The consumption of fatty acids in the brain is at least tenfold slower than other tissues. Okay.
Hmm.
So we're talking slow. The other thing is buffer, and we haven't talked about the adipose tissue, right? Because let's say you eat a bunch of DHA, I don't think you just send more to your brain, you send some of it to your adipose tissue. And this is actually really neat and important from nutrition is the term infant has started to accumulate DHA right in the last trimester, and they're born. If you look at a baby, they're pudgy, right? They're cute, pudgy little things, right? There's a gram of DHA stored in that body fat. So if they don't eat DHA tomorrow or the next day, that DHA gets released and is serving as a buffer, I think, for the brain. Where this gets really interesting is the preterm infant. Okay, so they've missed out some of the in utero brain DHA accretion into the brain because they came out a little bit too early. The other thing is if you look at a preterm infant, they don't have that chubbiness, right? They're a lot leaner, so they don't have that gram of DHA. And so we've got a lot of clinical studies with infant formulas that came through kind of the 80s and 90s that didn't have DHA in them and infant formulas that had DHA in them.
Mm.
And they tend to work well in that situation on cognitive scores. Okay. You can have better cognitive scores in a randomized trial like that.
So
You and
infants who
I,
had omega 3s in their formula had better cognitive scores than those who had formula that lacked omega 3s.
generally speaking, yes. Okay. And then there's controversy because you measure those cognitive scores at a fairly early age. Do they exist later in life? Sometimes yes, sometimes no. Are the effects big enough? Not clear. My colleagues in Australia just did a study in premature infants where they gave them DHA. They were actually maybe very premature infants, although I'm not the best with those definitions. They gave them DHA above standard of care, and they were able to show probably what we call a meaningful improvement in cognitive scores at 5 years of life. It was published in the New England Journal of Medicine, which is where, from a nutritional, if I can call it supplement study, that's a pretty cool place to publish it. It tells you something about what people think the importance of that is. Okay, so that's the infant which is trying to grow a brain and presumably also has that turnover, which I was alluding to, this 5 milligrams per day uptake. And then we get to you and I, and we've got a couple of things going on here. We've got the slow uptake, 5 milligrams per day. Our brains aren't growing anymore, right? We've hit that plateau, so we've lost that. We've got this body fat buffer that can have grams of DHA in it for some, but not for all people. And so we've got a situation where, and I don't want any of the people studying appetite regulation to get upset here, but if you live in my world, I'm not too worried about what I eat tomorrow, right? Because I think I've got a slow uptake rate in the brain, and I think I've got a buffer to get it there. But to your point, chronically over a lifespan, that's when we can change things. And in fact, if you take a rodent, we can even do this in a rodent. If you give me an adult rodent and I try and lower its brain DHA levels, it's hard to do. Sometimes we don't do it. So the way we do it is we rig the experiment. We start with a low omega-3 diet from birth when it's got to grow the brain, and we keep it on that for a long period of time. And that's how we change the DHA levels. And very importantly, we in that case, we lower them, right? We don't increase them. And so, yes, to your point, can we do this in humans, right? And we don't have great brain data, right? So we don't have a lot of you can't ask people after they died what they ate, right?
Yeah. What about
Yeah.
the rodent work? So when you do a study where you are, from birth basically giving them DHA deficiency, so they don't have enough omega threes. As they go through life, do we know the effects that has on the brain and the ability for those rodents to do learning and memory tasks?
Hundreds of studies, maybe not hundreds, but 100. And the totality of the evidence suggests a few things: impaired memory, impaired learning, impaired behavioral scores, more susceptible to neuroinflammation, more susceptible to a variety of things. It's a bad position for those animals. A lot of us are working on the molecular details, which involved a lot of these signaling molecules that we're talking about, and even more complicated, sometimes crosstalk between the omega-3s and the omega-6s. But I think the totality of the evidence, maybe there's 50 papers that have looked at brain inflammation. Generally speaking, most of those studies, they seem more susceptible to it. They seem more susceptible to neuronal death if you give them whether an ischemia model or a neuroinflammation model. And, I don't do a lot of behavior work myself, but my colleagues who do behavior show behavior scores in kind of baseline, especially in response to challenges in those animals. So pretty clear literature there.
Okay, so in rodents, you guys can give them chronic omega-3 deficiency, and basically bad stuff happens. There's more brain inflammation, there's learning and memory deficits, and there's no benefit that comes from being omega-3 deficient. Related to that, I want to ask you about, so you've got omega-6s, which we generally think of as being pro-inflammatory because they're used to make stuff that is used for inflammation in the body, and the omega-3s are typically associated with anti-inflammatory effects because they're associated with molecules that resolve inflammation. But then I'm interested in their metabolism, because from my understanding, they are often processed by some of the same enzymes. So this gives rise to the notion that the omega-3s and 6s are sort of competing with each other. And so based on what you just told us about omega-3 deficiency, which, scientists can induce experimentally in rodents, I'm wondering if you could have, is it possible to have an effective omega-3 deficiency, even in the presence of some dietary omega-3s, if that omega-6 omega-3 ratio is just highly skewed in favor of the omega-6s? And is that something that we should think about?
It's a huge debate, and it's a challenge for us to study, because when in nutrition and in membranes, when you lower something else compensates, right? So a fair criticism of the interpretation of our studies, especially the ones that haven't dove into the mechanism, is we lower omega-6s in the brain, omega-3s in the brain, and we raise omega-6s, and we kind of ignore that. And we say, oh, this is a DHA effect, right? And in some cases, I think we have other, you can get into cell culture models, and so the DHA can recapitulate some of those effects you see, or a lack of it, you can. But there clearly is a side of this cascade, as you mentioned, to the omega-6s. In some of those mediators start to become more abundant. And again, I think I told you this doesn't make a lot of sense to me, right? Because we still have enough of the omega-3s in that membrane that they've dropped 20, 30%, but not 99%, right? So they're still there, but we still see these shifts from the omega-3 mediators to the omega-6 mediators, and you gave kind of a summary of them there. That explains some of these results we're seeing. And some people, although in the brain, I don't think you know, the brain's neat from a homeostatic perspective. I can change an animal's blood levels of omega-3s, probably in a couple of liver a few more days. But the brain in a rodent, we're talking a month, right? At least. And that's why we do it premature. So a lot of my colleagues, I think, slip up a little bit and look at a study and they see these massive effects of the balance and these kinds of things in the liver and the blood and some of the white cells. But we don't quite see that in the brain because it's got this added level of homeostasis, probably because the uptake rates are low, the consumption rates are low, and we've got a few buffers to get them there. So I think you're right. It's just a slower process than people might think it is sometimes.
In terms of, so if omega-6 fatty acids tend to be pro-inflammatory, what do we know about the consequences of chronically high dietary intake of omega-6 fatty acids in terms of what the brain's doing, effects on learning and memory, effects on inflammation and things like that?
Yeah, so not as many studies, okay? And I know there are people who think they know the answer to this question. But it actually gets pretty tricky because it was actually hard to make a diet, because that's how we do this, right? We don't use knockouts, at least not until recently, that was low in omega-6s and not also low in omega-3s. So if you go back into the old literature and somebody's saying, they were doing all this in the 50s, 60s, 70s, this kid, which I would love to be called, doesn't know what he's talking about, right? You read those papers with a little more detail. They were what we would call essential fatty acid deficiency. And so that's a new term. And it means they were deficient in omega-6s and omega-3s. And if the title of the paper says omega-6s, it's probably, with a couple exceptions in the literature, misleading, right? So that created a hiccup in our field in that we thought we were studying omega-6 metabolism, but we were also studying omega-3 at the same time, okay? And people realized this. One of them was my old PhD advisor, that there was some nuance there. And we studied this a little bit more detail. We haven't done nearly as much in the brain, though, okay? I could probably count on my hand the amount of animal studies that have been done to intentionally lower omega-6s without lowering omega-3s and measure outcomes related to that. But I think it's broadly consistent with your position. I'm saying lower, you're saying higher. I think I told you it doesn't matter which way it's the same coin, two sides. But if you get the higher omega-6s in the brain, you get increase of those mediators, you get some of these effects. And then consequently, you tend to get lower omega-3s in the brain too, and you get lower of those omega-3, resolving signals, which is a really fascinating area as well. Okay.
So in
not
general,
nearly as much work.
Higher omega-6 does tend to translate to more inflammation.
Yes, in the brain. Now, so there are exceptions, right? Not every mediator that comes from these omega-6s is pro-inflammatory. In fact, as I understand it, when they're in the lab that eventually won the Nobel Prize, one called lipoxin, one of the molecules called lipoxin came out of this arachidonic acid cascade, and they noticed it was actually anti-inflammatory, and that confused them, I think, at the time, because they'd all been pro-inflammatory. And that postdoc then went on to discover most, if not all, of the anti-inflammatory pro-resolving molecules that I think you alluded to. And I just find that a fun story because he was exposed to a molecule that didn't make sense in the model, and then was very open to discovering the rest of those molecules, right?
Given that we know that some of these fatty acids have a role in promoting or resolving inflammation, given that we know that some of them serve as precursors or act themselves as neurotransmitters, and they play into like endocannabinoid signaling and things like that, I would expect that these fatty acids and the composition in our diet they're going to have effects on things like mood and cognition. What do we know about any sort of mood disorders or psychiatric disorders where either a fatty acid deficiency or imbalance is thought to be important for the cause, or whether an imbalance is important, say, for the progression or the severity of the disease?
Okay, so great question. So a couple ways to look at this. One are the animal models. Works well in the animal models, as you'd predict, right? If you take an animal and you remove the omega-3s, which is a lot more research done on it, these can be models of depression, aggression, behavioral learning, et cetera, et cetera. Okay, so the animal models work. You know probably better than I that an animal model depression is a little controversial. Let's just say
Mm-hmm.
that, right?
Yep.
So how does that extrapolate to the humans? Well, let's look at the humans and see what we've got. We've got some postmortem studies that have looked at DHA levels in the brains of, say, depressed patients. Literature is not clear. Somebody can show it, somebody can't show it. They don't all have the same regions. It's a really tricky thing to do, but that's what that is. And then, well, what do we do? How do we test this? Well, we give them DHA in their diet to see if we can make their symptoms better. In general, the answer is no, it doesn't work. But there's two buts to this. It's kind of hard to do because of what I was telling you about that slow uptake, right? So if you knew that the uptake rate was 5 milligrams and that there was 5 grams, and you thought the depressed patient had 4, you could guess how long you should do that study for. But if you do a 3-week study and you say, oh, it didn't work, it didn't work. There are other nuances in these studies that make them very complicated to do, right? So I'm going to go on a little bit of a tangent, if you don't mind, because I got involved in a clinical trial on
Mm-hmm.
epilepsy. Same idea. Let's give them fish oils. Let's see if it works. So if somebody comes in with epilepsy, first they go see their neurologist and they try and treat them with the drug, right? Hopefully that works and they can't enroll in your study. Okay, they're out because they've been treated. If that drug doesn't work, the neurologist will try them with another drug and then at some point another drug. And eventually, if they don't respond, then they'll come into your study. Okay, so now you've got a special population here that's not responding to drugs. And ethically, the neurologist keeps treating them, okay, while you're trying to do the study. And this, as somebody who came into the clinical world as a little bit more of a naive basic scientist, and now I'm in a faculty of medicine, these studies aren't exactly what I'd like them to be for a variety of reasons, right? We're trying to treat people with refractory epilepsies who are being medicated, and the physicians are changing their medications while we're doing the study. So it's not what we thought. And depression's not quite the same thing, but it's a little bit like that. But then what we've got is this wild finding in depression that there's, for reasons we can get into if you want, but people, one of the problems with fish oils is they're not drugs, right? So why would a big company want to do this study? And then they've been able to patent derivatives of fish oils, especially one with this EPA. And if you do the studies, you kind of don't see anything, but if you go back and do a meta-analysis and do what's called a meta-regression analysis and do it by the composition of the oils, you see that some of the oils that contain EPA show promise, show amelioration of depressive symptoms. Very few studies have looked at prevention because that's hard to do, right? You need more people to do that. Maybe one study has looked at prevention and it didn't work. So there are little signals in there, but there's two ways of looking at this. One is if you're a nutritional scientist or if you're a psychiatrist, you'd say, well, the effect's not big here. And if you have to do it, it takes a really long time. So I'm not going to treat my patient with this because it's going to take 1,000 days and I got to treat them. Fair point. If you're a true nutritionist, you'll say, haha, but we should prevent this. And then we haven't done all those studies, right? So one of the advantages nutrition has is you're not gonna take an anti-epilepsy drug or a mood drug just in case, right? You're gonna take it when you're diagnosed. But nutrition is something you got to decide to do, and we can do it just in case. And unfortunately, we don't have those studies, so I can't tell you for sure. What I can tell you is correlational studies, they tend to work, right? So people who eat more fish tend to have better outcomes in this. And we can talk about Alzheimer's as well going forward with that. So, and then the biologist in me or the neurochemist in me says, well, sometimes I don't really care because these molecules are pretty cool. And I think the molecules are doing things that are really important and they're working in the animal models. And whether you can regulate them nutritionally or not, I want to take a pharmacological approach with these and move that field forward. So,
Mhm.
so there's a variety of ways to looking at this. Bottom line, I don't think nutrition or lipid therapy is going to cure depression. There's some room for some signals and maybe a way to help us understand it. Prevention's completely understudied, and we should probably do something about that, right?
I see. So is there very much known about neurodegenerative diseases and whether or not adequate, having adequate fatty acid in the diet or having deficiencies in omega 3s or something else can make disease progression happen faster, even in animal models?
Yeah, so animal models again works pretty well. And there was a study published in Neuron, geez, it might be about 20 years ago by Frederick Cologne and Greg Cole, where the first Greg Cole was working on some of the earlier animal models, and Frederick Cologne was a little interested in nutrition, and they put the two together and it worked really nice. So these animals with higher DHA were protected, and a lot of people, I assume even myself, have replicated those studies. So the animal models works pretty well. But you know as well, there's somebody sitting there saying, so do a lot of the drugs work in the animal models that don't work in the humans, right?
Mm-hmm,
It's
mm-hmm.
like we're not supposed to talk about that sometimes, but that's a big one, right? And then, so what do we have in the humans? Well, in the neurodegenerative diseases, there are a handful of studies, not all, that can postmortem show lower levels of some of these lipids in the brain. Great. But they're neurodegenerative diseases, and if you have neurons dying, you might expect a bias in the lipid composition. So we got a chicken and egg issue, right, to some extent. Um, we've got clinical trials, pretty good ones, and pretty large studies in Alzheimer's disease. Yeah, I'm going to say it doesn't work. If someone wants to disagree with me on that, they can go ahead. Then we get into the like more the MCI phenotype and this, and my take is it generally doesn't work. People do subgroup analyses, post hoc subgroup analyses, and they can show you some signals and maybe a subset of people who had the most mild forms of it. And that's a little controversial. Some people are going to say, aha, it worked in those people. We should do that. Some people say, no, no, that just tells us we got to do a new study with them a priori. And then sometimes the nutritionists come in and say, these aren't drugs, people. Your standards are too high. It's diet. The the brain healthy diet is also heart healthy. So let's do this as a preventative measure and see what happens. And lastly, if you look at the epidemiology, like the fish eaters have lower incidences of some of the neurodegenerative
Mm-hmm.
diseases of the Alzheimer's type
Mm-hmm.
kind of thing. So there's some evidence there.
Yeah. Given what you've told us about a lot of the stuff, the slow turnover rate in the brain of some of these things, the buffering. What's your opinion on like fish oil supplements and whether or not those are very effective or, as effective as just eating, a whole food diet like that contains fish and things like that? Is there like I don't know, for the average adult in America, is there actually much risk for true omega-3 deficiency? Can the supplements really help? What do you think about that general world?
Yeah, so the last case of true omega-3 deficiency I know of was published in the 80s. A young female had a gunshot wound in her stomach area, and they put her on an intravenous feed for whatever reason, didn't have any omega-3s, and she developed neurological symptoms. They added soybean, I think, to the intravenous feed, and her symptoms disappeared. So we don't have that omega-3 deficiency syndrome around much in the population. Uh, my colleagues would be screaming at me right now because they say nobody cares about that. What we want to know is there a level that would prevent or help with neurodegenerative diseases, not these old case studies from the 1980s and 1960s. Fair point, okay? And so sorry, I've lost my train of thought a little bit right now. If you can remind me, we were
Yeah, I mean, are there
oh,
any? Is someone like
supplements
truly at
versus
risk? Yeah, do the supplements work? Are we
yeah.
truly at risk for omega-3
Okay.
deficiency as just normal people walking around today?
Okay, so let's walk through these one at a time. We'll go forward and then we'll go back a little
Okay.
because they circle back. So fish intake and Alzheimer's disease. no studies done, right? No randomized studies that have done this to try it, at least not to prevent it. But we can use the epidemiology, and we can see that people who consume more fish have a lower incidence of Alzheimer's disease. Fish is a little expensive. What city are you in? Sorry, I can't remember what city
Seattle.
you're
I'm
in.
in Seattle.
Yeah, so you got fish, but I'm sure you got beautiful fish markets.
Yep.
It's expensive. It's expensive though, right? And I'm in Toronto, and same thing. And so the problem with that is we worry that there's some sort of confounding that fish eaters have better health care, and you
Yep.
can
Yep.
go
Yep.
all. So it doesn't show causation. Fine. Should you eat fish? Well, looks good. But the counter argument is, well, if I'm not eating fish, what should I eat? Should I eat pork? Should I eat a beef burger? And I think fish comes up pretty high when you get into those kinds of arguments. Okay, and then you walk along, you say, well, I what about a fish oil supplement? Okay, should I take that? And I think the randomized controlled trials of fish oil supplements aren't overly strong as well in the neurodegenerative disorders. Maybe it's too late. Maybe we can't fix them that far. The drugs don't work that well, right? In fairness, so why would a fish oil work? And then people say, well, what about prevention? What about, I just don't like fish. I don't need to eat it. So now we're going to circle back. Okay. So when you eat fish, and this is where nutrition becomes a disaster. It's really fun, but a disaster. You eat a piece of salmon, which has got that nice level of omega-3s, or tuna, seared tuna. You've also chosen, if you look at that plate, you may be having a glass of white wine. You may be having some vegetables with it. You might be squeezing a lemon on it, right? And so this is where the fights come in. Somebody says, oh, it's the antioxidants in the lemon that's actually beneficial. And somebody says, no, it's the fruits, the vegetables you're having with the plate. Wine 20 years ago, they would have said it was it, but now that one's a little more controversial. And then somebody says, no, it's not that. It's what you're not eating. Who would have a fresh tuna steak with French fries and potato chips, right? So you're displacing things. So the mechanism of action of food is probably very complicated, right? And then you say, well, I'm going to take a fish oil supplement to get around that. And I'm going to go to the fast food restaurant tonight because I had my fish oil supplement. So it becomes a bit of a tricky situation. Fish oils, the omega-3s are similar to that we find in fish. The absorption is roughly similar, and they'll change your blood levels that are roughly similar. So if you purely argue that omega-3s are the sole mechanism of action, you got a reasonable point, okay? But I'm not convinced that they're the sole mechanism of action because of the complexity of nutrition, which is so complex now that sometimes we call these dietary patterns. And you see that they can go to a grocery store in some countries, and based on whether you bought fish, they can predict whether you're going to buy olives or you're going to buy a soft drink, right? And because people eat in patterns. So I kind of didn't answer your question, but hopefully the nuance and some of the difficulties came through.
Basically, it sounds like an omega-3, a fish supplement, an omega-3 supplement will work in the sense that it's going to get into your body. It's going to be akin to actually eating something like fish. The question is, are the omega-3s the reason why we see some of these associations in the literature, like lower levels of Alzheimer with fish eaters? We don't know if it's the omega-3s per se, or just eating a healthier diet overall, or what have you.
Exactly. I agree with that. Yeah.
Okay.
And I'll be clear, the associations are strong.
Yeah.
And sometimes very consistent, right? So I don't want to understate that. And reasonable people will look at that literature and say, I've got to eat something, and this is the best case scenario, right? Like I don't want to throw foods under the bus, but eating potato chips does not strongly positively associate with protection from Alzheimer's disease.
Right,
it just doesn't.
Yeah.
So I get that decision.
Well, I mean, kind of related to that, there's a lot of people who are concerned about seed oils and the high omega-6 consumption that we have in the modern world. People will point to things like, if you look at pre-modern humans, they were maybe approaching a 1-to-1 ratio of omega-6 to omega-3 intake. Biologically, it kind of makes sense that you'd want those things balanced because one of them is pro-inflammatory and the other one's for resolving inflammation. Obviously, you don't want too much inflammation. You just want to use it where you have tissue damage or where you have an infection. So a lot of people make the argument that having a really skewed omega-6 to omega-3 ratio, which most people do today, 10-to-1, 20-to-1, even 30-to-1, that's generally going to be bad because you're going to get more inflammation. Are you concerned at all about the level of dietary omega-6 intake today, and how do you think about that?
Yeah. So yes and no. There's a bit of a problem where I think some people have gone a little too far with the data we currently have, and I think we need to get more data. So one of the things I think people can do is that they can use what's called ecological epidemiology. Okay. And this is an approach where either you can look at different populations or time, right? And you can say Alzheimer's incidence has increased over the past X hundred years, so is seed oils. Bingo. Okay, got it. Okay, that's some sort of evidence. I don't think it's particularly strong evidence. We can do a better job with what we call prospective epidemiology. And these are studies where we characterize what people eat at an individual level, so not at a population level. We're not comparing the incidence of Alzheimer's in Japan to the United States, right? We got individuals all in the same country, and we follow them for a period of time. And we don't have as many of these studies because I think the field of nutrition was largely driven by cardiovascular disease. So these big cohorts were set up to study cardiovascular disease, and that's what they were designed to do. And a few of them are now starting to say, hey, let's look at Alzheimer's in these things, right? And so seed oils do not show up, as far as I can tell, as a massive smoking signal or gun or issue in those types of studies yet. And in fact, in cardiovascular disease, they show up as a bit beneficial, okay, in those kinds of studies. And then people lose their mind and they say things like, oh, nutritional epidemiology is not good, blah, blah, blah. Fine if you want to take that perspective, but then you can't argue this ecological stuff with me, right? Because we're going down the level of evidence. So you want to set a bar here. I'll respect your logic, but let's speak above the bar, right? But I'm open to the idea that there's biologically plausible mechanisms at play here, okay? So this pro-inflammation, these oxidized lipids, and now that some of my colleagues are saying, hey, you can absorb some of these things, especially if they're rancid and stuff. So I'm like, I'm not sure what the answer is. I've got a grant submitted that got rejected to study some of this. So I think, I'm not saying, I'd never say there's nothing there. But clearly people go a little bit too far. I think, the neurodegeneration is going to be hard to study, right? It's just a hard thing to study from a nutritional perspective because of the exposure time. So let's see if we can do what I would call the proof of concept studies and see if it makes a difference and pathways, biological pathways that we're concerned about in major depressive risk or Alzheimer's disease. We haven't really done that yet. And I think we need to be careful, though, just saying there's oxidized lipids in the Alzheimer's brain, there's oxidized lipids in seed oils, draw the line, right? So there's a few steps in that pathway here. So I think it's reasonable that we should take a look at this, especially in long exposures. It's going to be difficult to do. I don't get too excited about it personally yet.
So, I mean, just given that you study lipids generally, has this impacted you at all in terms of your life, in terms of your fat diet? Do you eat a high fat diet, a low fat diet? Do you pay attention to the particular fats you're consuming? Are there any general principles there that you think are pretty solid for people?
Yeah. So there's a few things with me. I'm in a nutrition department. If I sit down on an airplane or at a dinner table and people ask me what I do, I say I'm in the faculty of medicine because I don't want to talk about it, okay? Because nutrition can get a little too exciting with some of these things. So some days I'm a neurochemist and my family might look at me and I thought you said you were a nutritionist, right? And some days I'm a nutritionist. So I avoid that. All joking aside, yeah, has had a big influence on me. One thing that you wouldn't know about me personally is I'm a bit of what I would call a foodie. So I'm into things like that. And I think that drives my dietary pattern a little bit. But one of the things I recognize about diets is that, we don't have the best evidence at hand. We don't have randomized control trials on all these individual things. And we've got mixed data on some things. In general, the the epidemiological data for the brain, with some minor exceptions, that looks healthy for the brain looks like it's good for the heart. And I'm happy about that because it would be a real disaster if there were two different diets, right? And so these diets that are high in that are plant-based, not necessarily vegetarian or vegan, which we can talk about a little bit if you want, that have a little bit of a lower content of meats, red meats, and have fish, fatty fish in them, look like they're pretty good for cardiovascular disease, and they correlate with the brain. Sometimes things like blueberries and show up a little bit more in the brain side than the cardiovascular side, so it's not all that consistent. So
But
yes,
you're talking about epidemiological literature.
yeah, yeah. Let me take a step back. Then we've got a handful of, and some people would say this isn't true, but we've got a handful of randomized controlled trials on cardiovascular outcomes, okay? And roughly speaking, these Mediterranean style diets look like they work in those studies in cardiovascular disease. So we got an epidemiology on cardiovascular disease, we got an epidemiology on the brain. They correlate, okay? We've got randomized controlled trials of diets in cardiovascular disease. We don't really have them in Alzheimer's disease, okay, of diets. We've got supplements, but not diets. So I'm missing that part of the equation. So what do you do? Well, I think it's pretty reasonable to say, like, this stuff that we see for the heart looks like it's also pointing in the same direction for the brain with some factors missing and some important factors, right? So uh, I'm, I, some of my colleagues are really interested in ketones, not an area we went, but are a byproduct of fat oxidation. And one of the ideas here, I think that's really important, and we need to study this, is that in the Alzheimer's brain, there's a reduction in glucose uptake. Why is that? Well, you got dead neurons, so less glucose is coming in, okay? Sure. But some people are also saying, oh, no, 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 there's a little more than that. And if you look at it with a little more nuance, it looks like the glucose impairments, the glucose uptake impairments are early on. And so they're wondering if they can bypass that with other brain energy sources. Proteins are tricky because the proteins pretty much don't cross the blood-brain
Mm.
barrier.
But
Amino
ketones work.
acids are amino acids are regulated at the transport level, but ketones you can get in and get them through. So they're working on this, and we spent the whole talk, and I avoided that aspect of fats. But can you rescue brain energy deficits with ketones? Good point for the brain. And then that puts us is into a little bit of an area where some of that, the way to increase your ketones might not be ideal for your heart or for some other things, right? We don't need to go down there. So there's some little nuances to go through. But yeah, I happen to live near a fish shop. I pop in once or twice a week. If I'm in a rush, a can of sardines, it makes for a great snack with an apple. Nutritionally, there are a lot of other things I think you can do, trying to keep your waistline a little tighter than a little bigger, which is nutrition and some other things. But if you looked at my diet, you'd say this is a guy who eats Mediterranean-ish with a lot of exceptions on the weekends or when he's traveling or doing things, right? Because of the foodie side of me, and I like it, right? I enjoy the fresh stuff coming in the season. So.
Are there any like major misconceptions you feel that are out there about lipid metabolism generally, or brain lipid metabolism in particular, as it relates to diet, or things where people seem to be very confused?
Yeah, tons of them. Uh, One is if you Google or ask AI, I do this in lectures, what the most abundant fatty acid in the brain is, it'll say it's DHA, and it's just wrong. Okay. And
Mm.
we walked through that earlier. But DHA's become this dogma. It's the most abundant fatty acid in the brain. I love this molecule. I'm fascinated with this molecule. I think about this molecule, right? I love it. So I don't want to downplay it, but it's not the most abundant in the brain. What that has done is it's led to maybe a dogma that ergo the dietary requirements of it are quite high. And we didn't get into this. There's also a huge debate because the precursors that you can make it from are what we would call plant-based. It's not a totally accurate term.
Yeah.
So some people say if you look at the rate of that reaction, it's not that fast. and you got a lot in the brain, so they say you can't make enough, so therefore you have to eat it. But for me, it's a little different. It has to do with how much is coming into the brain, not how much is into the brain. So there's just this fundamental understanding, and DHA is very important for the brain, but how much dietary DHA we need to consume to do that, it's not clear to me. And at one end of the spectrum, some people say it's a really, really high number. Okay, so I think that's one of the dogmas that's there. The other thing is I think that, and it's true from a maybe a behavioral perspective, and if you eat fats, it'll determine what you eat, how much you eat at your next meal. And so there are behavioral aspects with this, but I really don't think we have any evidence that we can meaningfully change brain fatty acid composition in the short term. Okay. It's hard to do in animals. I suspect it's going to be extremely hard to do in humans. So that's another area that I think is an issue, that the people look at these things in terms of very short timelines. When nutritionally speaking, we're thinking of probably long timelines, which evolutionary is very important, right? Because if you had times where you couldn't get access to these things, the last thing you want to do is have your cognitive abilities dropping when you're searching for food,
Yeah,
right?
yeah, yeah.
That would not be good, right? So we've got preservation mechanisms on these things. But then that's not to say that there's not something there that could decrease the incidence of some of these disorders in the long term. It's just these are hard studies to do, right?
Hmm. All right. Well, we've already covered a lot. This has been fascinating. Is there anything you want to leave people with? Any final thoughts? Anything you want to reiterate from what we talked about lipids and the brain in particular?
Yeah. I would just say, we talked about fatty acids, and any lipid chemists coming on here said we forgot about cholesterol, we forgot about ceramides, we forgot about these things. Why did we do that? Because we talked about the ones I study. That's what I do. But it's a really fascinating field, and I think it's really growing neatly. So we've had advances in people have probably heard of all these omic techniques, metabolomics. Well, lipidomics is blown up now, and let me just throw one little story at you that
Hmm.
kind of gets one of the problems with studying lipids as signaling molecules in the brain is these phospholipases. Okay, so one of them is called calcium dependent cytosolic phospholipase A2. The calcium dependent might give you an idea what goes wrong here. So if you try and take a brain, well, am I saying this right? Out of a rodent, almost any way you do that, you're going to cause ischemia, you're going to cause hypoxia, you're going to get this glutamate signaling, you're going to get calcium release, and you're going to activate these enzymes post-mortem,
Mm.
after death, right? And then the fireworks go off. These mediators show up that don't even exist and weren't there, and they all change. So we have to be very careful to the person who's listening to this saying, I'm going to try this out, about how we collect these tissues,
Yeah.
because
Saying that the
the
very way that we
signals
collect these tissues is going to change the lipid profile that you measure.
complete, completely, right? And so we're in a neat time right now where we've got, I think the improvements in molecular biology and the neurosciences have been fascinating. And I think lots of the things we were talking about where we can't knock out a lipid, we can come really close now because we can knock out the enzyme that synthesizes it or degrades it, right? And so we're finally, after decades of struggling with these things where we remove it from the diet or not, and we change the whole body, not just the brain, right? And then we blame the effects on the brain. We're getting these tissue specific, cell specific knockouts, and we're starting to make these advances with these changes in lipidomics. But every so long we take a couple of steps forward and we take a step backwards, and some of the methodologies on lipids aren't as intuitive as some of the other fields. So I think it's an exciting time. We're also getting advancements in imaging brain lipids, which are going to take us quite forward. And people are just interested in nutrition. And so hopefully we can do more of these studies. Part of the neurodegenerative epidemic might be related to the benefits we've seen in cardiovascular disease, so people are just living longer, right? We didn't have Alzheimer's that much in the early 1900s, 1800s. People are living longer. So we're getting there, and I think we've got a lot of fun tools to study this field.
All right. Well, Dr. Richard Bazinet, thank you for your time, and I hope to talk to you again at some point.
Thank you very much. It was an absolute pleasure to be on and talk in such detail. I'm really glad you're doing this.
This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.